If you want to be successful at the Aurora and get the highest quality data from it, then it's critical that you bring high quality reference controls for every single experiment. I have found that the most common cause for getting poor quality data off of the Aurora is that you brought poor reference controls. So in this video, I'm going to give you 5 rules to follow in order to produce these reference controls. And I'm also going to tie that in with making the unmixing algorithm work properly, because that is ultimately what the purpose of these reference controls is. If you have not watched the previous video where I go through the basics of the unmixing algorithm, you might want to go back and watch that video first before you watch this one. Now, I will give you a lot of detail on all 5 rules to produce excellent reference controls, but in general, these 5 rules are going to consider these different factors that I've listed here. So we definitely want to make sure that we're recording enough events for every control, that they contain both a positive as well as a negative signal. We need to be aware of the autofluorescence properties of our reference controls because that factors into how the unmixing is calculated. We need to pay attention to how bright the fluorophore is relative to the full stain. We also need to pay attention to the signature of the fluorophore relative to the full stain. So now I'm going to jump into all of the details for all of these considerations. So rule number 1 is that reference controls must have sufficient events in both positive as well as negative populations. If you are running compensation beads, 5,000 events is probably a good number for your bead controls. You will get sufficient positive and negative events. If you are running cells, it's a little bit more complicated than that. If you have a marker that's like CD3 on PBMCs, then I would recommend somewhere around 10,000 to 20,000 total cells. If you have a marker that is more rare than that, so the frequency of the positive population within the entire tissue is more rare than CD3 in PBMCs, keep in mind you're going to have to record more total events so that you get enough positive events. So you might need to record 50,000 total cells. Also pay attention to if you have debris in your sample. That could change the number of cells you have if you have the cytometer set to 20,000 total events versus 20,000 total cells. I'm also going to comment about the unstained control here, although all of these rules that I'm talking about are actually talking about the single stained controls. But in terms of running enough events, I also want to mention for the unstained control, you should run enough unstained cells. It's going to be utilized for autofluorescence assessment. So you should keep in mind that you probably need to run more events than you're used to running for your unstained control, especially if you're using the autofluorescence extraction feature. If you have a homogeneous population, like a cell line, then you might actually be okay with somewhere around 10,000 to 20,000 cells for your unstained control. However, most people are going to want to run more than that, especially if you have multiple cell types within your tissue. Keep in mind, you probably want to run, I'm going to ballpark it somewhere around 50,000 to 200,000 events. It really depends on your tissue and how many different autofluorescence signatures you have within that tissue. It's just something you're going to have to play around with. Now, you're probably wondering, how do I actually judge when is enough events? So the software will give you a minimum. I thought it was 200, although it might be 300 events. But somewhere around 200 or 300 events, when you draw the gates on your positive population and your negative population as you are unmixing your data within the software, that gate must contain a minimum of 200 to 300 events. If you don't meet the minimum, the software will give you an error message and you will not be able to complete the unmixing. I will also say you shouldn't be aiming for the minimum. You should aim for more than the minimum. And that goes back to averages. So if you recall back when you were a child and you learned about how averages work, an average of 200 total things is going to be not as good as an average of 2,000 total things. So in terms of cytometry and events, I would probably aim for somewhere around 1,000 to 5,000 total positive events of similar fluorescent intensity. And you'll notice here the spectral plot. So when you record your reference controls, you'll view a spectral plot, which shows you a density plot of all of the cells within the positive gate. The algorithm doesn't want the signature from all of the cells. It wants a single signature. So it's going to take an average of all of the events and come up with a single signature that will be utilized to calculate the unmixing. You need to run enough total events so that you have roughly 1,000 to 5,000 positive events in your reference control. Rule number 2 is that positive and negative events should be clearly separated. I honestly think that this isn't the most important rule to follow, but we should pay attention to it. So you might think that that means having a pattern that looks something like this, and that is certainly a great situation to be in with your controls. However, if your marker is not an on-off type marker and it's maybe more of a continuum, and you have a range of intensity for your positive population, that also is going to follow this rule quite well. You're going to gate on just the brightest events. So having this type of marker is exactly the same as having this type of marker as far as what the algorithm wants. If you are in this type of situation where I have the unstained cells in the gray histogram and the stained cells in the red, I think you would be able to get the algorithm to work. It's probably not the best situation to be in, but you should be able to calculate unmixing. However, if you're in this last situation here, this is really not something that you would want. And if you are in this situation with your control, I would definitely consider alternatives. Rule number 3 is that the positive and negative events must have identical autofluorescence properties. This rule relates to how you set up your controls within the software and might not be so critical about how you actually stain your reference controls. So the reason for this goes to how the unmixing algorithm works. Again, you're going to be drawing a gate on your positive population as well as a gate on your negative population. The algorithm is going to assume that within this positive gate, there are 2 signatures. There's the signature of the fluorophore as well as the signature of the autofluorescence. In the negative population, this contains the signature of the autofluorescence. So the algorithm just wants the fluorophore signature. And in order to get just the fluorophore signature, it's going to subtract the negative signature out of the positive signature. And that will give it the signature of just the fluorophore alone. So in order for this math to work properly, you need to make sure that the autofluorescence between the positive gate and the negative gate is matching. If the autofluorescence is not matching, then the math will be thrown off a little bit. Another way that the math might be thrown off that is related to this has to do with compensation beads. Please do not be lazy like I used to be and just stain the compensation beads and then run them on the cytometer. You're going to need to wash those beads before you run them. If you don't wash them, then your negative beads will actually get a little bit of non-specific staining. So you'll see fluorophore signature, and that will throw off the calculation for your controls. Now, in terms of your cells controls, considering the autofluorescence of those, you might end up in this type of situation. So in this example, we have CD14. It's stained on human blood, and our CD14 positive monocytes are here, and we don't really have any CD14 negative monocytes in this tube. So that means when we draw our gate on our negative population in the CD14 reference controlled tube, that negative gate is going to contain lymphocytes and neutrophils, and the autofluorescence of these cells is going to be different than the monocytes in our positive gate. So in order for the autofluorescence properties to match between the positive and negative particles, we need to have an unstained monocyte population. So we need to set this up in the software using a feature called universal negative. That requires us to bring an unstained sample or unstained tissue. That means if we have multiple tissues within our experiment, we definitely need to make sure we have multiple unstained cells. I will put a link in the YouTube description box that details how you actually set this up in the software and more considerations related to the universal negative feature. Rule number 4, I would say that a lot of people mess up this rule. The positive signal intensity of the control has to be as bright or brighter than the multicolor sample. I have 2 examples here. On the left, the signature is dim, and on the right, it's the same exact signature, it's just brighter. So if we have this left signature and this is our reference control, and we give this information to the algorithm, and then we run our fully stained sample, and it looks like this right situation, then the algorithm is going to have to extrapolate some of the data, and it will find the signature, but you're going to end up with some unmixing errors because it's extrapolating data. If we have the opposite situation, so if this signature here is our reference control, we tell this to the unmixing algorithm, and then it looks for that signature within our sample and it ends up looking like this, then that's very easy for the algorithm to find the signature in the sample and determine exactly what fluorophore channel it should be placed in. One more note about this rule is you actually don't want your control to be too bright. You don't want it to be several logs brighter than your sample. So this signature on the right here is about the brightest you would ever want one of your reference controls to be. The peak detector here, the intensity in that detector is at about 10 to the 6. So that's about the brightest you would ever want any control in your experiment to be. If our signature for our reference control is too bright and is basically off scale, then we might need to adjust the instrument settings, and that would have an impact on our fully stained sample if that data was much dimmer, and that just makes everything very complicated. So it's okay if the control is brighter than the sample, but you don't want it to be so much brighter that you have to modify instrument settings. Rule number 5 is another critical one that I see a lot of people messing up. Fluorescence spectrum of the positive control needs to be absolutely identical to the one in the multicolor sample. There's also special considerations based on what type of fluorophore you have. If you have a tandem dye, for example, APC Cy7, PE Cy7, PE Dazzle, you'll notice they have dashes in their name. In that case, you must be sure to use exactly the same antibody, even exactly the same lot. The reason for this is when the manufacturers make these tandem dyes, there's no perfect chemistry to say put exactly 10 Cy7s on every single PE molecule. So when there's a new batch of creating tandem dyes, it could be a little bit different. The signature could be a little bit different from batch to batch. So we need to make sure we're using exactly the same lot to get the best unmixing results. An example of that would be if you have samples and controls that you've run on day 1, and then you come back on day 30 and you have run the same panel and want to reuse your controls from day 1, you need to be sure that you use the exact same lot of antibody. If at some point between day 1 and day 30 you purchased a new vial of antibody, then you're going to have to bring a new set of controls. There's other considerations you need to make if you are using old controls, and I'll link to a video in the description box that goes through what you need to consider. But with your tandem dyes, you definitely need to use same lot. For non-tandem dyes, your basic ones like PE, APC, FITSI, BV421, you could potentially use a different antibody that has the same fluorophore. An example of this is maybe you are staining tetramer, which typically tetramers have either PE or APC, and it's probably pretty difficult to get 1,000 tetramer positive events for your single stain control. In that case, because the fluorophore is not a tandem dye, we could use a different antibody that has the same fluorophore. So if our tetramer is PE, then maybe we use a CD4 PE antibody for our reference control just to unmix the data. Another reminder, please do not use equivalent fluorophores for your controls. Some people have picked up these bad habits where they use a FITSI single stain control for their sample that contains GFP, or maybe they use a BV510 control for their fully stained sample that has the live dead aqua dye. You should not be doing this ever, but it is particularly important on the Aurora that you do not make these swaps. Please use the exact same dye for your controls as you use for your fully stained samples. There are some special considerations if you're using a lineage or dump channel. That means that you have more than one antibody with the same fluorophore, and you're going to use that dump channel to remove cells that you're not interested in looking at. In the panel design video, I'll mention how there's a few fluorophores that are kind of annoying, which make them really great for dump channels. For example, PE-Dazzle 594. This is a tandem dye. If you have this situation, what is not recommended to do is buy 5 PE-Dazzle antibodies. Instead, the proper way to do this, according to Cytek, is to buy 5 biotinylated antibodies, and then you come in with a secondary streptavidin so that the same tandem fluorophore binds to all of the antibodies within the dump channel. And finally, the last consideration is that fixed perm buffers have the potential to alter the spectra of a fluorophore. So you have to keep this in mind when you are preparing your reference controls and make sure that your reference controls are exposed to all the same buffers as your sample. Later on, we'll talk about some staining conditions. The only buffer that you don't need to keep the same between your reference controls and your sample is a polymer stain buffer. But all other buffers, every other treatment that you do to your sample needs to also be done to your controls. Even if it doesn't make sense, you probably do not need to permeabilize your compensation beads. However, we need to make sure that the antibodies that are bound to compensation beads are exposed to the fixed perm buffer in the same way that our samples are exposed to the fixed perm buffer. Now, quick quiz, just to make sure we are all on the same page. Let's say we have this antibody against PDL1. We've got the fluorophores PE-Dazzle 594, that's our sample. And then for our reference control, we're going to use the same exact antibody and we're going to stain the same exact cells. Will that unmix correctly? Hopefully, yes. This is the one that should always work, assuming that everything is the same between the control and the sample, same cells, same concentration, same antibody, same everything. This should always unmix correctly. Now, let's talk about the same sample, but instead of cells, we're going to use the antibody on compensation beads. Should that unmix correctly? In theory, yes, but sometimes we run into issues where actually this does not work out so well, and I'll talk in a couple of slides about why that is. Okay, third scenario. Let's say we're staining FOXP3, that's on BV750. And maybe I decided that it was kind of hard to get a good positive staining for that marker. And so I wanted to swap that out for a CD4, also on BP750. Will that change work and will it unmix correctly? This is the one where I tricked you because I did not tell you all the tandem dyes. So the brilliant UVs and the brilliant violets are sneaky. There are a lot of tandem dyes that don't have dashes in the name. So the ones that are not tandem dyes are the first ones of the series. So BUV395 and BV421, also BV480 and 510. Those are the only non-tandem dyes in this group of fluorophores. All the other ones are tandem dyes. So that means that BV750 is a tandem dye. Because it's a tandem dye, I am not allowed to swap it out with something different. And you can check out our website. I have a list that you can download off of our website that will list, from what I can tell, what I've researched, which fluorophores are tandems and which ones are not. Okay, let's talk about this scenario, which is we have a tetramer, and that's on PE, and we decided that is really hard to get enough positive events, so we swapped that one out for CD4 PE for our control. Can I make that switch? Will it unmix correctly? That one should be okay because PE is not a tandem dye. And the last one, we have this activation marker CD69. That's on APC. Our fully stained sample has some activated cells in it. And for our control, we have the same antibody, and these cells we didn't do any extra stimulation to. Will that work? Probably not, because most likely, this being an activation marker, and since our sample is activated, but our reference control is not, most likely the marker will be dimmer on the reference control, and because of that, that will not unmix correctly. So keep in mind that you might need to manufacture your reference controls if you're using cells for them. That means maybe you need to stimulate your cells. Maybe you need to use a different tissue or cell line that has higher marker expression. That also means if you are using a different tissue or cell line for any other reason for your reference controls, you need to be aware of the level of expression of your marker on that tissue compared to your actual sample. Because if it is a lower expression than your actual sample, it's going to be a problem. There is one control that you will definitely need to manufacture at all times, in my opinion. That is your viability stain control. You definitely want to make sure that you have dead cells and live cells in that control. If you don't have both, it can be very difficult to use. So my recommendation is to actually kill some cells. There are lots of ways to kill cells. You want them to be dead, but still intact. So you don't want to kill them so much so that they're basically exploded and just a bunch of debris. So the way that I tend to do it is I'll put about 100 microliters of cells into a Eppendorf tube, pop that in a heat block at 65 degrees for 10 minutes. Then I shove that tube into ice. That works really well for me. Another way to do it is you could try just leaving your cells in the fridge overnight, and you'll get a decent percentage of dead cells. You don't need tons and tons of dead cells. You just need a very clear population of dead cells. Once you do that, most people like to use the fixable viability stain. So you'll stain with that, then wash that away. And then if you want to, right before, you can spike in some unstained cells if you're not sure if you still have live cells left. Now, the topic of compensation beads versus cells. Because a lot of people hear about these rules for reference controls and think, that's going to be hard to do on cells. I might as well just use compensation beads and those should work really great. Unfortunately, that is not the case. And in some cases, compensation beads are quite challenging. So here's an example of the same signature, the same antibody. On the right, we have it stained on compensation beads. On the left, it's stained on cells. And you can see the signature looks a lot better on the beads. What I mean by that is that it's brighter and it's cleaner. There's less variability. So the variability is determined by how much red there is. So there's more variability on the cells than there is on the beads, which it is what it is. But in general, signatures on beads tend to look really great. However, there is this weird quirk that we don't fully understand. It's definitely something that is known to happen. Unfortunately, it's very unpredictable as to when it happens and when it does not happen. But for some reason, sometimes when antibodies bind to beads, they have a slightly different signature than when they are bound to cells. And if the signature is slightly different, that will lead to unmixing errors. So I'll show you some examples of that. So if we overlay the two spectrums on top of each other, so basically taking these two plots here, putting them on top of each other, unfortunately, there's no quick way to do that. We've got to export the MFIs for all the detectors and then graph them in Excel. We've got to normalize to the peak channel. So regardless of brightness, you would expect that the signature on the beads, which is in blue, and the signature on the cells, which is in orange, should be perfectly overlapping. And you see that in this region, right around R4, there is a mismatch. So if we take the single stain beads and we unmix either single stain beads, which is this top row, or single stain cells, which is this bottom row, and we check how accurate the unmixing is. And again, to check the unmixing accuracy, it's the exact same as you would check compensation accuracy. So you want to be able to draw a perfectly straight horizontal line right through the middle of the positive and negative populations. So we can see that the beads unmix the beads perfectly. This looks great. But if we look at how well the beads unmix the cells, it did not do as well. Particularly this one here does not look great. It looks like it's overcompensated. Instead of compensation, we're doing unmixing. So we generally tend to say this looks overcorrected. And we can actually predict this based on this plot because the main mismatch is in the R4 detector. Fluorophore that peaks in R4 is APC R700, and that is what is on the y-axis here. So this is APC R700, and then this is BV711 for the BV711 single stain cells control. We can also see the same thing for per-CP eFluor 710. There's multiple mismatches, and the beads unmix the beads correctly, but the beads do not really unmix the cells that well. So what does this mean for your real experiment? If I see somebody bringing an experiment where they only have compensation beads for their reference controls, they don't have any single stain cells, they've never run any single stain cells previously to test out their panel, this is a big red flag for me because you can end up in this situation where your controls are going to look like everything is perfectly unmixed, but then when you go to your cells and you try to unmix your fully stained cells, you're going to have unmixing errors, and that's probably going to lead to some false positives, and you're going to be getting incorrect information from your data. So the only way to correct any unmixing errors is to have single stain cells or to do some panel checks and figure out where this mismatch happens. Again, the mismatch is very unpredictable. So just because this particular per-CP eFluor 710 antibody has this mismatch doesn't necessarily mean that every single one you purchase has a problem. I've also heard that you have to check for this mismatch with every single vial of or lot of compensation beads that you purchase. So just be cautious if you are using compensation beads. And I don't want to fully discourage you from using compensation beads because they can be really helpful, particularly when you have low expressed markers. So here for this BV480 sample on cells, this is what the signature looks like. And I've also shown you the autofluorescence signature for these cells. So if you can imagine subtracting out the autofluorescence signature, there just really isn't a whole lot of BV480 on these cells. So it's going to be kind of hard to unmix using this particular control. However, if we use compensation beads, we're going to get a much better signature to provide to the unmixing algorithm. And that's probably going to be the way to go in this case. What I would recommend doing is while you're setting up your panel, do one experiment where you bring a double set of controls. So let's say you have 10 markers in your panel plus a viability die, so 11 total. So I would recommend for one experiment, you're going to bring single stain compensation beads for all 10 antibodies. You're going to bring single stain cells for all 10 antibodies plus your viability die, and then you're going to bring your fully stained sample. And we're going to use these controls to figure out what combination is best to successfully unmix your sample. And you're going to assess how successful the unmixing is using the single stained cells. So you can try different combinations. You can try unmixing with just the compensation beads. You can try unmixing with just the single stained cells. Or maybe there's a combination of both compensation beads and cells that gives you the best possible unmixing results. Once you have figured out what your best possible combination is, then you can just bring that combination, that single set of controls in order to unmix your data properly. And I do have a video for precisely how you would determine all of this. Lastly, people ask me what compensation bead should I use? Does it matter? I don't have a perfect answer for if it matters or not. I personally have only tried beads from Thermo Fisher, and I will tell you that out of the Thermo Fisher beads I have tried, the UltraComp eBeads Plus are the best ones from them. But there are lots of other manufacturers that make beads, so you don't necessarily have to use these ones, but these are the ones that I have found work.